Today I'll be going on linkage and crossing over. So generally when we talk about linkage, what it means is it is the tendency of a genes to remain together. So there are a few examples. The first one is a cross in Drosophila. These are nothing but your fruit flies. So generally what happens is the genes for gray body color and long wings I have designated as capital B and capital V. For your black body color I have designated as small b, and vestigial means short wings I have designated as small v. So these are the genes that are responsible for production of your gray body color, black body color, vestigial wings and long wings. So generally when we talk about linkage, let's just do a simple cross. This cross or these characters are basically observed in your flies that are nothing but your Drosophila. So a cross between black vestigial and gray long flies. Black vestigial I have designated by small letters. So these means these are the recessive characters. Gray and long I have designated by capital. So this means these are the dominant characters. So the gametes are So in the F1 hybrid, we're getting an individual with gray and long wings. They are a hybrid because they contain both sets of genes. Now what I'll do next is I'll be crossing this F1 hybrid with a double recessive parent. So these are the gametes I'll be getting from the hybrid individual. So these are the offsprings that I'm supposed to get when following the law of independent assortment. But in reality, when I'm crossing a gray long with a black vestigial, what I'll be getting is only gray long and black vestigial offsprings. These are the only offsprings which you will get. That means we are not getting gray vestigial and black long. So if we are not getting this offsprings, that means we are not following the law of independent assortment. And this also provides a clue that the genes for gray, that is capital B, and long, capital V, they are linked together. That means they do not tend to separate, because if they separated out, we would be getting this type of offsprings, that is gray vestigial and black long. So this one was the expected ratio, that is we are supposed to get gray long, gray vestigial, black long and black vestigial, but in reality what we'll get is only gray long and black vestigial. So this proves that the genes for your gray and long they are linked together. So this is what it means by linkage, the tendency of a genes to remain together. They do not separate out during gamete formation. So this example is also an type of complete linkage. That means the genes, they are completely linked. We do not get any sort of intermediates between the main offsprings. We get only gray long and black vestigial. Now the next type of linkage is incomplete linkage. In incomplete linkage, you will get slight deviations from the independent law of independent assortment. You will generally not get the always 9:3:1 ratio. This is the normal ratio that you're supposed to get when you're following the law of independent assortment. But in incomplete linkage, we do not get this ratio. There will be slight variations. So let's just do a simple example in sweet pea. When we are crossing a flower with blue and long pollens and a flower with red color and round pollens. These are the genes that are designated. Capital B represents blue color, capital L represents long pollen grains, small b represents red color, small l represents round pollen grains. So normally what do I do is I'll cross a blue long with red round. So obviously the genotypes will be So in the F1, what I'll be getting is a hybrid individual with blue color and long pollen grains. Now, this F1 hybrid, again, I will cross it with a double recessive parent. Your double recessive parent is your red color and round pollen grains. So when I'm crossing a hybrid blue long with red round, I'm supposed to get blue long, blue round, red long, and red round in the 9:3:3:1 ratio if I'm following the law of independent assortment. This is the expected ratio that I'm supposed to get, but in reality, what I get is the observed ratio is 7:1:1:7. That means I'm getting 7 individuals that are blue long, 1 individual with blue round, 1 individual with red long, and 7 individuals with red round. So this ratio is quite different from the expected independent assortment ratio. So this proves that the genes are incompletely linked. Most of the time, the genes for blue long, that is capital B and capital long L, they tend to remain together. They separate out only once. That is, what I'm getting is blue round. I'm getting this combination only once. So this shows a tendency of a genes to remain together, not completely but incompletely, because I'm getting different combinations like blue long and blue round. So this shows that the genes, these are not completely linked. They also tend to separate. So that's why I'm getting different combinations. So this is an example of incomplete linkage. So it's because of linkage that we get slight deviations from the law of independent assortment. The next thing is crossing over. Crossing over is one of the most important phenomena which leads to different combination of genes. Crossing over generally takes place during meiosis. So just take a look over here. Crossing over means the exchange of chromatids between two homologous chromosomes. In this case, one chromosome I have represented with white chalk and the other chromosome with green chalk. So during crossing over, what happens is these two segments of the chromosomes, they exchange with each other. As a result, this new chromosome has a different combination of genes. Because of crossing over, we get different sets of characters. So this is an example of single crossing over. That means there's only one exchange of chromosomes. This is an example of double crossing over because there are two exchanges of chromosome arms. So generally, because of crossing over, it is possible to get different combinations of genes. So the thing to remember when we're dealing with linkage and crossing over is the distance of genes that are responsible for characters in the length of an chromosome. If the genes are close between themselves, that means there is more chances of linkage. If the genes are far away, that means there is more chance of crossing over, because this segment might break over here and it might break over here. But even if a break occurs, since they are very close to each other, let's say over here, the genes are very close together in this chromosomal arm as compared to this arm. So even if a break occurs, these two genes, they always remain together. So as a result, they undergo linkage. The sex of an individual is determined by sex chromosomes. In the humans, there are two types of sex chromosomes, X and Y. For a male, the chromosome combination is XY. For a female, the chromosomal combination is XX. That means you're getting one X chromosome from the father and the other X chromosome from the mother. But whereas in the case of male, the Y chromosome is always obtained from the father, the X chromosome is obtained from the mother. Males are heterozygous for XY, females are homozygous for XX. So we'll be continuing with this in the next class.